Okay, let's get started. It's a pleasure to introduce Professor Yaniv. He's an associate professor at the Weizmann Institute, where he also did his PhD, followed by a postdoc with Mark Schnitzer at Stanford, looking at calcium imaging and behaving mice. And he's going to talk to us today about his exciting work looking at neural representations in the hippocampus.
Thank you. Thank you very much, Aya. Can everybody hear me like that? No? Great. Excellent. So I will So I'll talk today about representational drift in the hippocampus and cortex. So in my lab, we are interested in neural coding of long-term memory, and specifically try to understand how memory information is encoded in the brain and what happens to it over timescales that range from days to months. And specifically, the approach we take to do that relies heavily on optical imaging, specifically time-lapse calcium imaging, which we perform in freely behaving mice using miniature fluorescence microscopes and microendoscope probes and genetically encoded calcium indicators. Now, this approach, which is, I think, pretty in common use right now, has many advantages for neuroscientific studies, but for us, for the purpose of studying neural coding of long-term memory, this approach offers two critical advantages. The first is the ability to record from a fairly large population of neurons, more than 1,000 neurons simultaneously, and this is important because it allows one to analyze the data not only by accounting for the activity patterns of individual neurons, but also to account for the joint activity patterns. And the other key advantage, of course, is the ability to track the same neurons over many days, weeks, and months. And this is important because it really gives us access to how neural codes evolve as a function of time and experience. So this is an example of a typical data set that we obtain in the lab. You can see here the same mouse running back and forth along a simple familiar linear track on 2 different days of the experiment, while we image neuronal activity in its hippocampus CA1. And if you pay attention here in the middle pane, you can see and those 6 example cells where the mouse is located with this cell fire. And of course, these are not just any cells that we picked at random. These are hippocampal place cells, so cells that encode the position of the mouse within the environment. And if you pay attention even closer, you can see that their firing is directional. So they prefer to fire in a specific position, but only in one running direction. And when the movie will stop playing in a few seconds, you will, I hope, be able to appreciate that the neuronal responses were kind of pretty much retained stable across these 2 days of imaging. Now, when we were first managed to get the system up and running, the first question that I was interested in was to what extent neuronal representation of such familiar environment is stable or dynamic over time. And we did a very simple experiment, like I showed you now, where mice were running back and forth along a linear track and were reintroduced to this familiar linear track every several days, specifically every 5 days throughout a month. And what we found was rather surprising. We found that hippocampal representations of this fixed familiar linear track gradually change with time, despite no changes in the behavior, in the visual, other cues, anything that we can control for. And the basic finding is as follows. So here you can see the map that was formed by all the places that were active place coding on day 5, the first day of the experiment. And you can see what the same cells were doing, were coding later on. So this is after we aligned the data. So you can see already 5 days after, many of the places that were coding on day 5 are not active or not place coding on day 10. And you can see that this specific representation kind of fades away with time. but this is not something that is unique for the first day of the experiment. If we take as a reference day 20, the middle of the experiment, we can see this gradual buildup and decay of the representation. And if we align all the data according to the map that was formed by all the places active on day 35, we can see that most of the places active here are actually not placed coding in the beginning of the experiment.
How do you know it's not the neuronal, the stability of the experiment, of the identification of the neuron?
How do we know that, sorry, that what?
That it's not you lost the neuron.
Okay, so how do we know it's not a problem in the registration of the identity of the same neurons over time? So we invested a lot of work into identifying and benchmarking it, and you can see this. We can dive into this later. We can dive into the different controls that we did later, but this is well controlled for that, for cell registration. And yeah, so I would like to mention in this regard is that this phenomena that we now call representational drift is different than mere variability in the neuronal activity, in that the similarity between any two representations gradually decline as a function of elapsed time, and this is not something that just variability in the neuronal activity leads to. And since we found that, several other groups have found similar results, similar phenomena, similar representational drift in the hippocampus, in different subfields of the hippocampus, using different techniques, both 1-photon, 2-photon imaging, electrophysiology, and also outside of the hippocampus using different techniques. So we know today it's more prevalent than we thought. Now, so I mentioned in the beginning we are interested in long-term memory, and this kind of phenomena is quite puzzling because, and specifically in the hippocampus, because we know the hippocampus is important for long-term memory of places and events. And so my lab has been really interested in understanding this phenomena of representational drift and its relation to memory. So today I will discuss some of the question we address with respect to this big question. The first thing is what might be the functional relevance of the continuously changing hippocampal representation, functional relevance to the function of the hippocampus. How do time and experience, or the absolute passage of time and the amount of experience, affect the stability or the drift in hippocampal neural codes? What is the relationship between hippocampal place code stability and long-term spatial memory? And to what extent representational drift is a unique property of deep or associative brain areas such as the hippocampus, the posterior parietal cortex, and others. So first of all, the functional question: what might be the functional relevance of representational drift? So again, this is the basic result that I showed you in the beginning. Now, the hippocampus is important for episodic memory, which you can think of as a memory of what happened where and when. Here we have, by definition, we have here information about where things happen. These are place fields, maps. And we ask ourselves, can this also infer the temporal relationship between experienced events? So this was the hypothesis that we wanted to check, that hippocampal ensemble dynamics, or drift, timestamp experienced events in long-term memory. And to address this, we did a simple experiment that is very similar to the one I showed you just now. Only now we require the mice to run back and forth in two different familiar environments. The environment were the same length, but otherwise we try to make all the cues, everything that we can control for, very different. And we imaged the mice every 2 days in both environments. One environment was imaged in the morning, the other in the afternoon, 4 to 5 hours apart. We switched the order of them so as not to be affected by any circadian effects or time of the day. And the rationale here was simple. We thought that if timestamping exists, it would not only help to dissociate between events that occur at the same place but at different times, like visiting environment A on day 1 and then again on day 9, but it would also help to associate events between between to associate events that occur at different places but close in time, like visiting environment A and B during the same day. But before we could do such an experiment or such an analysis, we need to verify that the hippocampus indeed represent these two environments very differently. And indeed, this is what we did. Oh, yeah. So this is what we did. So the fact that we are using two different environments doesn't in itself necessarily mean that the hippocampus regards them different, but it does. So environment A has a unique representation that is very different than environment B, and vice versa. And the similarity between them, which is quantified here by a measure I will mention it several times in this talk, the population vector correlation, which is the correlation between the population of between the activity of the entire recorded population per different beings along the linear graph. So this is very low and remain low throughout the experiment. And given this, we now calculated the similarity between the ensemble activity patterns in different sessions and different days.
Yaniv?
Yeah.
But these were two different sets of neurons because one had 590 neurons and the other one had 530.
In different environments, they can have a different number of neurons that are active.
So when you calculate the correlation between two population vectors, what do you do with the residuals? Or you are looking at the correlation between population vectors within the same.
That's a good question. I think you take the vector that is for the activity in both that consists of all the neurons for both environments.
Intersection between the two.
Yes.
Okay.
Yes. And by the way, because this is a line and you can correlate it like that or like that, we take the optimal, the higher of both. Okay. So what you can see here is that the representation of an environment to itself is always higher. The similarity between the representation of an environment to itself is always higher than the other environment, and this is what gives this checkerboard pattern that you see here. Also, that the similarity between the representation of environment to itself gradually declines with time. It's summarized here in the green curve. But also that the similarity between the representation of a given environment and the other environment also gradually declines with time, suggesting that hippocampal representations of these two different environments co-evolve with time. And given this, we asked, can we actually measure the we asked to what extent these dynamics of the two environments can infer the time in which different events occurred. And to address this, I will only show you what I think is the most exciting result. We did a decoding analysis, only unlike in typical studies of the hippocampus, instead of decoding the position of the mouse within the environment, here we decoded the time in which events occurred across the 8 sessions of the experiment. We trained the decoder on data from one environment and used data from the other environment as test data. We wanted to identify the day of the experiments in individual trials of 3 minutes out of 15. And we could do it accurately in about 60% of the cases. You can see that mistakes were for nearby days, and it's all very different than the shuffle control that sits exactly on chance level and more. Yes?
I'm a bit unclear about what you are doing with neurons who are place cells in one environment and not in the other.
Here I'm completely agnostic to this. I'm only, so.
You're using all the neurons.
All the neurons. And I think it's also impressive that if you take the day of the test data from the training set, so the decoder can only do the second best guess. This is indeed what you see. So in 80% of the cases, the guess is for the flanking days, and again, mistakes are for nearby days, which further speaks to how gradual this process is. So we thought this result suggests that experiences could be either associated or dissociated based on their temporal proximity and irrespective of where events occurred.
So neurons work in shifts?
Maybe not shifts, as binary on-off, but
No, shifts as in, we're working like in a hospital. We're now taking this day plus minus, and you'll take the next plus minus.
yes, but I think in a very smooth way.
So the take-home message here is that if I have a memory system where the memory decays, I can use the decay to repair when the memory began. The event began. That's always good to forget.
So the relation between this and actually memory is not so clear, and I will talk about this specific in several slides.
This is not memory.
What?
This is not memory.
Well, no, but you ask. This is you can think of it as memory, but you ask about the relation between. You ask if the decay of the memory in itself is.
Okay, so you're saying not memory. You're saying stability of the representation. It boosts that it decays because it gives you some information about time.
I think the similarity with the distance between different representations. Yes, this is what we suggest here.
Okay. So the brain has to store somehow the original representation in order to use it. You're using it because you know that.
So we don't know how. So, right, Chaim, if I ask you to retrieve from memory two events that occurred in the past, one 2 days ago and one 2 weeks ago, you think for a second and then you will recall. And the question is, how do you do that, right? What do you compare, right? And I agree, we don't know yet how it is done in memory retrieval.
Okay.
I would say that there is a study by Denise Kai and Alcino Silva's lab that showed consistent result using immediate early gene tagging, where they were able to associate and dissociate events in memory. This is behavioral level, right? Assessment over time scales of hours, like we showed here. So here, this is just neuronal activity level. They show the behavioral level. Memory level. Mickey.
Relates more to the previous result of the drift, but is there a difference between significant areas like the war zone compared to just any place on the, because the task here is kind of, they don't really need to remember where they are, except when they are.
Right. Yeah, keep this question in mind. I will talk about it in a few slides. But yeah, the answer is no. But I will talk about this. Yes. Okay. So right. So I showed you the mouse was running back and forth, and that things are changing over time. But we don't know from the experiment I showed you, they were just not designed to dissociate between the passage of time and the amount of experience that the mice had. So if we see a difference between the representation on day 1 and day 3, we don't know if the difference between the two is because they are 2 days apart or because they are 2 sessions apart. So we can address this question by basically doubling the interval between sessions, and now we can measure if we see the same difference between 2 sessions, irrespective of the amount of experience in between. This would suggest a time-driven dynamics. But if we see a similar difference for the same amount of experience and irrespective of the elapsed time between them, then this would suggest experience-driven dynamics. Clear? So Nitzani in the lab did this experiment, again using the same 2 different environments that I showed you in the previous experiment. So the same mice explored 2 different environments. One environment was visited every 2 days, the other every 4 days for a period of 3 weeks. These are familiar environments. And then we analyzed the data by accounting for 2 aspects of the stability of the code: changes in activity rates and changes in the tuning of the cells to position. So when we look on changes in activity rates and plot the data as a function of elapsed time, we see that the curve from the environment that was visited every 2 days sits exactly on top of that from the environment that was visited every 4 days, suggesting that the dynamics is time dependent. This is for changes in activity rates. And indeed, if we plot the same data now as a function of the elapsed sessions difference, and we can see the separation between the curves. So for example, for a session difference of 5, this translates into a 10-day difference for the environment that was visited every 2 days, and for a 20 days difference for the environment that was visited every 4 days. And you can see that the more elapsed time, the more dissimilar the code is. So this suggests that time is a more dominant factor than experience in driving changes in activity rates. So here we analyze changes in activity rates irrespective of changes in tuning, but we can also look on changes in tuning irrespective of changes in activity rates. And when we do that, we get the exact opposite. Here, when we plot the data as a function of the elapsed sessions, the two curves sit on one on top of each other, and you can see the difference when you plot the data as a function of the time difference. So actually, the more experience, the more change you see in tuning. Yes?
Does it matter if the additional experience is the same environment, or does it matter if the additional experience is the same environment? So if you compare the two A environments, when you have a B in between or not, does that matter?
You mean like that? Yes, so no. It was almost an invited question. No, yeah, exactly. So okay, so I'll go back so everyone can understand. So here in the timeline, you can see that there are two types of pairs of session in environment A, those without an intermediate experience in B and those with an intermediate experience in B. And you can ask whether this intermediate experience in another environment affected the rates of drift, and it didn't. So suggesting that experience-driven changes in spatial representation are context-specific.
The activity rate, can you explain? The neurons change their tuning.
So a cell can respond to the same place, but that just changes activity rate. One day it will fire in a higher rate or a lower rate in the same place. So the overall tuning curve will look very similar.
So you're looking at the same neuron,
yes,
different activity and the same position.
No, irrespective of position, just activity rate.
Position, axis.
No, this is an illustration to show you what we mean by changes in activity
I'm
rate.
trying to understand what you mean.
So just changes in the rate, the number of calcium events over time.
The neuron in a session.
Sorry?
The maximal firing rate of a neuron.
Not the maximal, the rate, the
average firing of the neuron in a session.
Yes.
Compared to the same neuron in a different session.
Exactly.
But why should it be different?
Why
Because
should it be
neurons
different?
will not-
I don't know. It is.
No, but what if, because neurons will not be responsive? I mean, you talked about stability of representation. Now you're saying something, you're talking about something else. If there is a remapping, in fact, a reorganization, so there is no reason that the neuron should have different firing rates. It's just a different position.
I think the way I think about it is that this notion of stability in the code, you can deconstruct it to two components. One is changes in or in tuning. So what are the actual inputs to this? And then the other is changes in activity rates. This can, biologically, such differences can stem from changes in the excitability of the cells. This can be intrinsic, extrinsic.
What I'm saying, the bottom one would predict that there is no change in it.
Would predict that there is no change?
In rate. Right?
No,
It just
this
different
just means
position.
that this is what I'm saying here, that the changes we look at here are irrespective of changes in rates.
If a neuron is firing in one session, then it has a place for the one session. And then in another session, it doesn't have a place for it at all.
Both of them will change, right?
Okay, so this is not the firing rate at the position of firing. This is just the average firing rate, and it relies also on the assumption that
This is the average firing rate, no position regarded at all. This is correlation in the tuning curve. So we've been the linear track. We divide the events along it. There is a tuning curve, and we just correlate it to cross section.
For the position, you don't even need a test, right? It's like overall time.
It is during the task, but yeah, but we don't regard the position here. Yes.
So when you talked previously about stability, which ones you would cover? Until now?
So in the previous talk, I talked about changes in activity rates.
Great.
Show the representation of space.
Right. So okay. So here, this is changes in activity rates. If you look on changes in tuning, there is also some change in tuning.
So if the place field is stable, except that some neurons stop firing, then this is what you mean? Neurons that fire at the same position. They're the same place cells,
Most of
except
the-
that some neurons don't fire. This will be the picture until now.
Sure. Yeah.
There is no remapping.
but
It's not
we see
remapping.
here
It's
that-
just neurons don't fire. It's not remapping because neurons that fire will fire the same position.
But they can change the firing rates.
Yes, I'm
They
saying-
still fire.
I'm saying the place fields will be stable, but it just, the maximum firing rate will be small, will be big or small. This is what the picture until now, which I think everybody thought that it's a remapping. That the neurons don't change their firing rate, it's just different position.
No.
Okay, good.
No, I don't want to go into this because this is well published, but like you can find many neurons like that are very stable over time. And if you look on the positional displacement of the centroid as a function of elapsed time, you can see that this is centered around zeros. Most of the cells.
What is zero?
Zero positional shift in the centroid of the place field across pairs of sessions
Okay.
with different intervals. So places, that's the thing. Places typically retain their place fields over time, but not always. They do change it. And we see what this shows is that experience, soon.
So other neurons will fire more. Some neurons will fire less.
Some neurons will fire less, some neurons will fire more, yes, but they also, to some extent, change their tuning. And what determines this change in tuning is the amount of experience, not the elapsed time. That's the bottom line here. Right. So, okay. So I showed you hippocampal representations gradually drift over days in a fixed familiar environment. The representation of different environments share time-varying components. That's the change in activity rates, maybe changes in excitability of the cells. Time and experience differentially affect distinct aspects of C1 code stability, and I think this is interesting because it may hint at different biological mechanisms that drive or restrict it. Okay, so until now I showed you mice running back and forth along a linear track without them needing to express memory. But in studies that do require mice to express or animals to express memory, the general assumption is that there are specific activity patterns that emerge during learning, and then during recall, the same activity patterns are reinstated, and this is what supports memory recall. And the general assumption is that the more stable the neural code is, the more stable the behavioral manifestation of the memory. And this is indeed consistent with experiments that manipulated neuronal activity over relatively short time periods between learning and recall, but the specific relationship between the two, between code stability and long-term memory, has not been measured. And this is what we wanted to measure here in the context of spatial memory. So to do that, Noah Sadeh in the lab trained mice in this more complex task, navigation in this 8-arm radial maze. So in this task, mice are released from, in each trial, they are released from the ends of one so-called start arm, and they need to navigate to the end of the 8th arm, the goal arm, where they get what are the words at the end. They do it based solely on distal visual cues. So here you can see an example of the performance of the mice during this task. This is on day one, and if you wait enough, you can see that how the performance looks like on day 5 when the mice are already almost experts in performing the task. And we had here 2 different groups, each group of 10 mice that underwent learning, training over 5 consecutive days. Then the so-called recent recall group returned to the maze 10 days after, on day 15, and had another session on day 16. And the remote recall group returned to the maze after 28 days of no visiting, and had another session on day 34. So this is the behavioral performance, and we see an improvement, declining the number of errors with learning, and then a time-dependent decline in memory performance after 10 days and after 28 days with rapid reinstatement of the behavioral performance.
So from day to day, what changes? Nothing? The goal is in the same place?
Yes, this is just spatial learning. Yeah.
I know,
It's
but
classical.
the goal is fixed always that arm, or there is a change in the goal?
The goal is fixed. What we do change within a session, every block of 7 trials, we rotate the maze on its axis to prevent the mice from relying on intra-maze cues like odor tracks and stuff like that. So this is to ensure that they rely only on distal visual cues. So it's really spatial reference memory. Okay, so the first thing we always like to do is to look on the coding properties of individual neurons, and what we find here are neurons that look like that. They start with a very unordered, I would say, or dispersed neuronal activity, which is refined with time, with learning, until you can see here typically a single field or several fields, but most prominently on one of the arm. And this specific place cell retained its preferred firing location across these 28 days of no visiting to the maze. And we can find other cells that look like that gradually refine in time, retain a stable place field across this almost a month of no visiting to the maze. So these are stable cells, but we can also find many cells that are also similarly refine their place field over time, but display a different place field after this month. So there is a heterogeneity in the tuning stability of place fields.
Is it common that in day 1 you see like very random, several locations?
Yeah. This is very yes. Yeah. By the way, there is also a change here in the behavior itself. So you have a diminishing coverage but you see the same thing when you just have another group of mice just randomly explore for randomly wards at the different arms. So the coverage is maintained. You see a similar refinement in the code over time.
Given this is by placing them in different arms at each point.
At the ends of the.
On their mistakes. You're inducing it by putting them in the arm you want to investigate, and then they quickly go to the rewarded arm.
So in each trial we put it in one of the ends of one of the arms, and the mouse needs to navigate to the goal. Then if we take it, we put it in an opaque bucket, like a biohazard bucket, just so it won't see. Then we bait the goal arm again, we put it in another, and we have a pseudo-random order for that.
So they see the goal, the visual cues
from everywhere. This is as
everywhere.
is. This is flat on purpose, so they can see everywhere.
So they have to learn to associate visual cues with the goal.
They have to navigate like we navigate in real world. There's no flag on the. So.
I understand. So
Yes.
it's the navigation.
Mm-hmm. Okay, now consistent with what I showed you here, if we quantify the amount of information individual neurons carry about the position of the mouse within the environment, you can see it's gradually increasing with time. Different colors at different times. We shift to the right here, and we can see a similar effect when we use a spatial decoder to decode the position of the mouse within the environment. We can see the decoder improves as the number of errors go down with time, and even when we account for the effect of time, we see a correlation between the performance of the decoder and the behavioral results, suggesting that memory performance correlate with place code quality. Now, what happens during memory recall? During recent recall, after 10 days, no changes in the amount of information, but there is a significant decline in how informative are the neurons about position after 28 days during remote recall. And what I'm showing you here for the pool of cells from all mice, you can see when we look, same thing when we look on individual mice, a gradual increase, then a decline after a month, and a rapid reinstatement. Now, what about the stability of the code? Here again, we use the same measure, this population vector correlation. So each pixel, each entry in this matrix shows you the correlation or the similarity between the representation of the maze at different sessions. And what we can see here is that if we now look on this secondary diagonal here, so compare the similarity between subsequent sessions, we don't see a stabilization of the code. This is summarized here. Despite the fact that the mice do improve the performance over time. And if we look on the similarity between the recall day and the last day of learning, day 5, we see a time-dependent decline in the similarity between these representations. Okay, so here I'm showing you the behavioral results again, and I hope you can see there's quite a bit of variance here in behavioral performance. There's quite a bit of variance here in the similarity of the code across time, and of course we wanted to see whether mice with a more stable representation would have a more stable memory. This is indeed what we see for the recent recall group. Here we correlate the population vector correlation between the recall day and the last day of learning with a change in the number of errors between the 2 days. We don't see such a correlation when we look on the data from the remote recall group. And this is not something that, just to show you that this is not something that is susceptible to our specific choice of a measure of similarity between representations. We did the same analysis now looking on the tuning curve correlation between neurons that were active on different days, and we see the same results. There is a relationship between code stability and memory performance during recent recall, but not during remote recall. So yeah?
So how can you control for the exploration of the behavior of the mice during recall? Because probably the color mode they may have.
So this is, yeah, good question. So here we took just the two first trials in the maze. So they don't have yet time to relearn during the task itself, not significant. So the first kind of reflexive response to these results from people who worked on the hippocampus is that maybe this is systems consolidation and the hippocampus suddenly becomes disengaged, not suddenly, but gradually becomes disengaged from the task after a month. But we don't think this is the case. First of all, Inbal showed already a decade ago that even in tasks that require the hippocampus, like contextual fear conditioning, even after a month, the hippocampus is still engaged and important for memory recall. Here we specifically chose a task, this navigation task that was shown to require hippocampus, intact hippocampus also during both learning and recall, remote recall. And also in our data, we see that even during remote recall, there is a correlation between the amount of information and memory performance, or between the change in the amount of information and the change in memory performance between the last day of learning and recall, suggesting that even after a month, memory performance correlate with place code quality. Okay, now I mentioned that we had another day of imaging on day 16 and 34. In this day, as I mentioned, we had this rapid reinstatement of the behavioral performance. Also, the amount of information neurons carry about position returned to its levels on the last day of learning, on day 5, and even exceeded it in the recent group. Now, if memories were stably stored in the hippocampus as a stable attractor states, then we would expect that this additional training on day 16 and 34 would cause the network to converge back to the same map that was established on the last day of learning. But if we look again on the similarity between the representations of day 5 and day 16 of 34 and compare it to that during recent and remote recall, we don't see such an effect. So both for recent and remote memory recall, both for population vector correlation and Unicode correlation, we don't see such a rebound to the previously established map. And finally, here, mice in the recent recall group, we also exposed them again to the maze on day 33. So here we can compare the behavioral performance. We see the behavioral performance was as good as it was during recent recall in the gray group. Special memory, special information, again, was similar to its levels during recent recall, but fast here, we see that the similarity between the representation here and day 5 was not higher as you would expect. Actually, it was not different than the one doing remote recall. So whereas recent memory retrieval helped retain neural code quality, the amount of information, and memory performance, it did not stabilize the representation. Okay, so there is a dissociation here. Code quality and memory performance goes hand in hand, but code stability doesn't during remote memory. So long-term memory is maintained in the presence of ongoing changes in hippocampal representations. Stability of hippocampal place code seem to be not a prerequisite for a persistent remote memory. And we think these results point to a population-level organization that enables retention of learned information despite ongoing changes at the single-cell level. Now, this work, yeah.
High population-level organization, if you can go into maybe the decoding is changing.
Maybe what?
Maybe your decoding is changing. Right, so you have your hidden link of dependency.
Sure, maybe
it's not the overall organization, it's the decoder that keeps changing as well.
By the decoder, you mean the downstream decoder in the brain? Yeah.
Because you need the retriever to
Right. So for that, the meaning of the activity pattern is changed, and the decoder has to somehow adapt to that.
Yeah, but you needed the retriever to keep using this decoder in order to keep the performance.
Yes. Right. So this is exactly what you.
No, but it's different than the hidden layer or the hippocampus needs to change to keep some kind of overall activity stable. Everything drifts, but the decoder keeps learning.
Right, and there is such a theory, by Tim O'Leary that suggests that the downstream reader may adjust together with the upstream circuit in a way that will adjust for that.
So it's not what you just said, because you said.
No. So let me clarify here. I think what we see here is that the standard traditional ways to analyze coding stability, in this case at least, fail to reveal to what extent remote memory is consistent over time. And this really got us thinking, is code stability, or the way we measure code stability, is the right neuronal measure to assess the conservation of long-term memory over time. And exactly as you said, the common methods for assessing the conservation of memory representation evaluate the stability of the neural tuning curve, but they rely on two critical assumptions. One is that if a memory is conserved, the same neurons will encode the same information over time. Not necessarily the case. And the second is that the identity of the encoded variable is known to us, such as the hippocampal neurons encode position. So we ask ourselves, can we relax somewhat these assumptions, and can we assess the conservation of memory based still on neuronal activity, but without relying on these assumptions? So Alon Geva in the lab developed a few years ago a new way to analyze neuronal population activity data. And I don't have time to dwell into all the details, but what these methods show is that you can quantify and describe the characterize the structure of similarities between neuronal activity patterns, and and the structure can be used to infer key properties of the internal representation, including reconstruction of the behavior itself. And this internal structure of neuronal population activity is different across different circuits, different brain areas, even for the exact same behavioral task. So here we compare between C1 and ACC. There's very different internal structure of neuronal activity for the two. But in the context of the discussion we're having now, what we found is that despite the ongoing changes in the activity patterns of individual neurons, this is again the linear track, the internal structure of neuronal population activity was conserved over time. So this is from the linear track. In familiar linear track, no behavior level memory here, but now we are applying the same analysis or similar analysis also on the data that I showed you now to test the possibility that the conservation of the internal structure of neuronal activity over time could better explain the persistence of memory than the stability of the coding properties of individual neurons. And we have some encouraging results from the learning phase in a somewhat different paradigm than what I showed you here. This is again in the radial maze. Again, here this is the structural relationship between activity patterns. So each data point here represents the activity of the entire recorded population within a single frame of 50 milliseconds. The color code is according to the arms of the maze, and I hope you can see the emergence of this structure with learning. And what we think is that, of course, this is still a work in progress, but we think that the same way that this structure emerges throughout learning, its stability over time may indicate the retention of memory. Yeah?
So how do you compensate for the difference in behavior?
So exactly. So this is what I mentioned earlier. Here we don't have this is a different paradigm. Here they forage randomly for reward in different different arms, and the coverage is the same. This is very important. Yes.
They move differently, about the different velocity.
We can control for all of that, but the behavior is very it's variable, but the variability is similar. Yes. There are things that change with familiarity, right? And
but don't they like alter shape, decide like very specific kinds of behaviors or
there are some behaviors that change with familiarity. For example, there is a scanning behavior that they do with the head and neck, and they do it less in a familiar environment, and we can control for the effect of that. But in terms of and they run a little bit faster. But other than this, it's very variable across mice. But it's the most important thing. You don't have a diminishing data coverage effect like you have in the regular what I showed you before. All right. So lastly, we ask: is representational drift is a unique property that is found only in deep or associative brain structures? And specifically, we were curious to know if representational drift occurs also in sensory cortical areas, and does neural code stability follow the hierarchy of sensory processing? And to address this question, we and by we I mean Daniel Ditch in the lab, took advantage of freely available data sets from the Allen Brain Institute, the Allen Brain Observatory data sets. This is several data sets. One consists of a 2-photon imaging data sets from tens of thousands of neurons in 6 different visual cortical areas across 4 layers, hundreds of mice that were head fixed and presented with different visual stimuli. The other data set is the neuropixels data sets, again from about 100,000 neurons in 6 visual cortical and 2 thalamic areas in tens of mice. The data, most of the data, all the data I'm going to show you now is actually from analysis of natural movies. So the calcium imaging data had, as I said, many, like tens of mice in each visual areas, and importantly had 3 different sessions that occur across different days. So it allowed us to analyze the data across days and timescales of days. The neuropixels data was one long session with different blocks of movie presentations that are separated 20 or 40 minutes apart. There are several data sets like that. And generally, it allowed us to see whether the same results that we obtain with calcium imaging we see with neuropixels. So in both data sets, we can identify neurons that respond to specific moments within the movie, the presented movie. Of course, I'm not suggesting that they encode time per se, just the specific scene that is presented during that time. So for instance, these 3 cells are from the calcium imaging data sets, have a very stable tuning in 3 different sessions. So each line here is a movie repeat. It's from the neuropixels data sets across 2 different blocks that are separated 20 minutes apart. So first thing that we analyzed, we used the neuropixels data set to analyze the data over timescales of minutes within a session. So on the left, you can see the population vector correlation matrix for different movie repeats in one example mouse in V1, and you can see this gradual decay as you move away from the main diagonal. And what you see here for one example mouse, you can see for all mice in all 6 visual cortical areas, you see this time-dependent decline in population vector correlation across timescales of 15 minutes. And we can see the same thing when we compare across different blocks, again within the same session, across all 6 visual cortical areas. And similarly, we see the same thing when we look on using the calcium imaging data across days. So this is again one mouse in area V1, and the population vector correlation matrix. You can see the same thing across all the many tens of mice across all the 6 different cortical areas. And what is nice is that they also record from different layers. And if we kind of pull together the data from the different layers, we see that drift occurred in all these different layers of the cortex. Different layers had different, I would say, intrinsic stability, but all of them display drift.
But for drift, you need to show us that you have an equivalent representation of the population, right?
Yeah, there is.
Did I miss it, or you?
I didn't show you, but like on each and every day, so this is day 1, this is day 2, and this is day 3. So you can see this is equivalent to this. So the secondary diagonal and the third are the comparison between.
But in neurophysiological data, we have tons of instability.
Sorry?
In the neurophysiological data, there's a lot of instability.
Yeah, no, there is instability, there is variability in the code, things are noisy.
Variability in the recording.
But variability in the recording, but you have some variability, but variability doesn't cause a time-dependent decline in the similarity between representation. It will just noise you the data.
Well, then you have accumulation.
What?
Then you can have accumulation.
Why? Why would you have a...
What happened to the tunnel?
I will show you.
The assumption that we can record exactly from the same cell for a very long time, that usually doesn't work in seconds.
No. Absolutely right.
So there is sort of, what you show here, the fact that it's so similar across layers, it doesn't have a lot of to do with the behavior. That suggests to me that this is like, type of problems in the recording. This is a how do you differentiate that between technical problems in recording? There is a slow change in the volume of the tissue or whatever.
No, this is an excellent point. And here we verify that the quality of the representation is similar at each session. Or across in the neuropixels data, we only used within one session, yes? So this was stable, and we used use different measures to show that the quality of the representation doesn't deteriorate with time.
But that's not the issue. The issue is that there is a slow change in volume of the brain that misaligns the neuron. Against the neuron. That's it.
So we I can show you that we did many control analysis to show that the alignment in the this is from the Allen Institute data. This is different sessions. We quantified the agreement using their methods of registration, using our methods of registration. So I think we are very confident about this. This is actually very clean data. But the most important thing, you don't have a deterioration in the quality of the representation with time. You also don't have an improvement with time. They are familiarized with the movie. This could also explain, right? Just learn.
But in your first, I think in the first set of results, you show that the amplitude of the response, the changes, the part, I mean, you have two parts of instability. One is the magnitude of response, the other one is the change in the tuning. The magnitude of response is something that might be related to this deterioration in the quality of the work.
Right, but it doesn't decline with time. It changes with time, but it doesn't decline, not monotonically, not monotonically. It doesn't decline with time.
I think that the question is, do you have like a positive example when you don't have, for example, if you split your data to two halves and you compare the representation on two disjoint classes of the data, then do you get like a flat matrix? Or what do you expect the level of change in representation while you do it within session?
So if I guess I didn't understand the first one. If you split the data.
I think the general feeling around is that some technical problem causes deterioration or changes in representation. There wasn't any example where there was stability. We never saw stability. Every time you compare two things, they're always kind of different to some degree.
But I can show you the stability within a session, right? So you have so everything here is normalized for the within session stability. First half, second half, or odd even.
So within session is 0.5. Is it good?
I don't know what good is.
So what is the complaint?
What?
You're showing it within a session. In this session also bad.
I don't know what's good and bad. I see a time-dependent effect here. For the, I don't have it here. This is kind of supplementary figure, but for the changes in activity. So also here we see changes in activity rates and changes in tuning. And for the changes in activity rates, we see them also when we select all the neurons that are just the neurons that display a highly stable tuning, okay, with a correlation of 0.6 and up, 0.7 and up, 0.8, 0.9 and up. Doesn't matter with the threshold. You still see similar time-dependent decline in the similarity between the activity rates of neurons, both with neuropixels and with calcium imaging data. Sorry, what I said now is just neuropixels, this analysis. So this is in neuropixels, so we know. So this is for you, Mickey. So there, I don't have it here because this is a supplementary figure. There you see that you basically take only those cells that display a stable tuning curve, and you still see a time-dependent decline in the changes in activity rates. Yeah.
Sorry for insisting about this thing, but I feel that the biggest problem, as Mickey stated, is that it would be really convincing to say that these cells do something in the same manner. It doesn't really matter, even spontaneous activity, but what you're answering, the fact that
you know, the population response was still carrying the same information or whatever, I mean, it's almost trivial that it will get less and less similar as time goes by, right? Because
Why?
your focal plane changes and
No.
tissue moves.
No, this you can control for.
I mean, how could you really control for that unless you show that there is something that these cells do the same, but the place coding does change? I mean, that would be the only convincing way to, right? I mean, that's also to be asked for. It doesn't answer the question that some of these cells responded stronger or weaker. The thing is that it's almost trivial that as time passes, we're less able to measure what we started with.
No, I don't agree that it's time. I mean, you could have a time-dependent deterioration.
You probably have always, kind of, whatever you mentioned. Right?
You could record from the home cage, right? Because presumably there are place fields in the home cage. Just have mice there for 10 weeks, and then you'd see, right, if the place fields in the home cage are stable or not.
Yeah, but even in the home cage, you will see a time-dependent change. So I don't know if this will address your question. So we can look on the same, same neurons, day 5 and day 33. So 28 days apart, and just look like identify same neurons within the same field of view, and even focus on those neurons where there is, I would say, for those of us who do imaging, there's little ambiguity that they are the same neurons, and quantify the tuning curve correlation between the place fields over days, and you can see cells here. These 5 example cells that have a high tuning curve correlation, cells that have low tuning curve correlation within the same field of view. This is just one example. We can quantify also the probability that, so you can put, if we look on pairs of cells from different sessions, okay, that are candidates to be the same cells, up to a distance of 15 microns. So you can look on the spatial correlation and the central distance with those cells. You can see all those cells that are here have high spatial correlation in the footprint, like this cell, and low central distance. These are likely to be the same cells. You agree? They must be the same cell. This is high spatial correlation. Low
You're
central
using only
distance.
those cells.
Very likely. What?
And you're using only those cells in your analysis.
I'm using all. I'm using no. I'm using right. I'm using only those cells in the analysis.
Only those cells.
Only those cells that we are certain that are the same neurons. Yes. But I'm just showing you here that this is during recent recall and this is remote recall, and this distribution are very similar. However, whereas this distribution are very similar, this population here, which we are pretty convinced that they are the same cells, okay, show a different tuning curve correlation, higher in the recent recall than in the remote recall. And I don't know if it addresses your question, Alan, but it's what we can do.
Maybe it's a good time to wrap up and we can continue the conversation.
Yeah, I think I have just one. I just wanted to show that even in the visual cortex, we see that this internal structure of neuronal population activity is retained over time, despite the changes in the coding properties of individual neurons. But so this is just.
What about Tala?
Ah, right. Sorry, you asked. Okay, so I will show this. So the question was. Does neural code stability follow the hierarchy of visual processing in the mouse? And recent studies by the Allen Institute and by others have established this hierarchy, where LGN is at the bottom, area LM is at the top, and we wanted to address this very basic question. Hypothesis that lower brain areas display higher stability, code stability than deep or associated brain areas. Or in other words, the closer you get to the primary sensory synapse, the more stable the code is. So we took, using the NeuroPixels data, first of all, the calcium imaging data, we looked on changes between V1 and downstream area LM, because they are adjacent to each other, so they're presumably it's less likely that we will be affected by any artifacts that have to do with the position of the, with any optical artifacts. And here, using both neuropixels data on timescales of seconds to minutes, minutes to days, and calcium imaging data on minutes to days, we see that contrary to the naive hypothesis, area V1 drifts faster than LM, downstream LM. And finally, for equations, when we using only the neuropixels data to look on differences between the LGN and the downstream area LP, again, two very adjacent structures, anatomically adjacent, we see that LGN drifts faster actually than the downstream area LP. Now, this suggests that the naive hypothesis that the closer you get to the primary sensory synapse, the more stable the code is not supported by the data, but I'm not suggesting that it's necessarily the opposite. Yeah, just
why are not suggesting it's opposite?
Because these are two pairs of brain areas, and there's not enough to show systematically that this is the opposite. Yeah, I think this is. I would love to look at the retina. Right, so I've shown you that drift takes place in multiple brain areas, time experience that fringely affect distinct aspects of hippocampal code stability. Long-term spatial memory is not maintained, or is maintained in the presence of ongoing changes in hippocampal representations, and the internal structure or the structure of relationship between neuronal activity patterns remains stable over time despite continuous changes in the coding properties of individual neurons. And yeah, and that's it. So these are the people who did the work. I've shown the work of Noah Sadeh on the radial maze, Alon Rubin, Daniel Ditch, the visual cortex, Liron Sheintuch, and Nitzan for all the linear track data. And really thank you for coming and listening, and hopefully we have